Hello and welcome to Legacy IAS Academy. In today's video, we are beginning with a fresh series in which we will be discussing the previous year's questions of prelims. Okay. So, starting with 2023 and with the paper of Science and Technology, we will be discussing all the questions which were asked in 2023. We will also be trying to understand why was this question asked, so that we can track down on what can be the potential areas of 2024's paper. Okay. So, beginning with 2023's paper, the first question which was talking about microbes, okay, microorganism. So, our idea is when we are discussing the prelims previous year questions and any topic has been asked, for example, microorganisms has been asked. So, we will try to take a 360-degree view of microorganisms. So, what are they, what are its types, what does it lead to, what are its uses, benefits, negatives and all that, and immunity and all that related to microbes. So, for example, they have asked a question on defense technology in 2023. So, we will be studying everything about defense technology. We will be studying about the integrated guided missile development program. We will also be studying about ballistic missiles, cruise missiles. So, just to ensure that next time if any question appears on missiles, we will not be missing out on those questions. That is the meaning of 360-degree you take. It is the meaning of taking a 360-degree coverage of every topic. Okay. So, with respect to microorganisms, the question that was asked was, we need to consider the following statements. Some microorganisms can grow in environments with temperature above boiling point of water. Usually we understand that if we boil water, then the bacteria, the microorganisms will be killed and the water will be safe for consumption. Okay. But then there are certain microorganisms which can survive much above the boiling point of water also. Okay. Boiling point of water also. The second statement says that some microorganisms can grow in environments with temperature below the freezing point of water. This is also correct. Although we have this assumption, we know this by fact, that boiling water kills the bacteria, kills the microorganisms and it is safe for consumption, or again freezing also. If you have to ensure that any object is pathogen free, so we basically freeze it. So, that is why we have, let us say, Norway may seed vault, okay, international seed vault, matlab all the countries are preserving their seeds. Why preserving their seeds? Because this is our understanding that at such low temperature we cannot expect any pathogen to survive. Okay. The third statement says some microorganisms can grow in highly acidic environment with a pH even below 3. Okay. So, what is an acidic environment? Acidic environment is any substance whose pH value is less than 7. Okay. If it is less than 7, it is called as acidic. If it is more than 7, it is called basic. Again, with respect to acidic environment, we know that any microorganisms usually do not survive in, that is the usual understanding, that they do not survive in such acidic environment. Kyunki putting up cleaning, for example, acids are used for cleaning. Okay, it is used for cleaning toilets, it is used for cleaning slabs, kitchen slabs. So, we make use of acids to ensure that all the pathogens on the toilets or in toilets or in kitchen slab, they are all cleaned, or on floor they are all cleaned, because usually in an acidic environment microorganisms do not survive. Okay. But then because they are asking about some microorganisms, so there can be an exceptional microorganism which can still survive in a temperature which is higher than boiling point of water or lower than the freezing point of water, or even a highly acidic environment or a highly basic environment also. Because there can be such a scope of microorganisms that we have not known that some microorganisms can be possible, it is possible for them to stay along these lines. Okay. So, the first statement is also correct, can survive above boiling point, can survive below freezing point. So, second is also correct, can also survive in highly acidic environment even with a pH below 3. Okay. So, this is also correct. Okay. So, in this question all three statements are correct. Okay. So, now let us start understanding about first of all what was the source of the question. Source of the question was NCERT. NCERT class 12th chapter 7. Class 12th chapter 7 Within the first introduction line itself, all these three statements have been mentioned, that there are some microorganisms which can survive above the boiling point of water also, below the freezing point of water also, and at a highly acidic environment also, highly basic environment also. This has been mentioned in the introduction of NCERT class 12, chapter 7. What are microbes basically, or what are microorganisms? Okay. Microbes or microorganisms. So, whenever we have this understanding that microbes will not be present above boiling point or below freezing point and all that, that was the usual understanding. We have this idea that microbes are usually non-beneficial. They are harm causing, they cause diseases, various diseases and infections. But then there are some microbes which are also useful. So, microbes are small microscopic organisms having either single cell or multi-cell. So, they can be unicellular or multicellular also. So, they can have single cell, they can have multiple cells. Then they can be prokaryotes also, they can be eukaryotes also. What are prokaryotes and what are eukaryotes? Prokaryotes matlab this cell will be primitive. Okay, primitive matlab it will have just single nucleus with no developed cellular structure. Or eukaryote matlab they will be having mitochondria also, they will be having nucleus also, plasma also. So there will be so many components of cell. Cell has evolved. But in case of a prokaryotic cell, cell has not been able to evolve. So the organism itself has cells which are primitive in nature. Okay? So prokaryotes are primitive cells, eukaryotes are developed cells. Microbes can be prokaryotes also, can be eukaryotes also, can be unicellular also, can be multicellular also, and a combination of these four also. So it can be unicellular prokaryote, unicellular eukaryote, multicellular prokaryote as well as multicellular eukaryote. Okay? Now talking about, so this is the meaning of microbes. Microbes ka matlab that only means that these are microscopic organisms. Okay. So what are the types of microbes? Types. So microbes can be classified as bacteria, okay, can be classified as virus, can be classified as fungi, can be classified as protozoa, and can also be classified as archaea. Okay. Amongst them, this organism, viral organisms, are considered to be an intermediate organism between living and dead. Okay. So if they are not a component of a living organism, matlab, for example, a person is infected by a virus, then because the virus is staying inside the living body, so they will be alive. As soon as this virus comes out, this becomes dead. Again the same virus, if it enters into a living body, any man, animal or plants, any body, if the virus enters into that body, then they will again become alive. As soon as the same virus comes out of the living body, it will become dead. So virus is an intermediate between living versus non-living. Okay? Rest every other thing are living. Archaea are also a form of bacteria only. Okay? They are a primitive form of bacteria. Okay. There is one more term that we need to understand here is bacteriophage. What are bacteriophage? Bacteriophages are basically virus. They are the virus which will kill the bacteria. This is the virus that kills bacteria itself. Okay. So the virus, this bacteriophage are the viruses which will kill the bacteria. Now this was considered usually, historically we have considered bacteriophage as a problem. Kyuki, let's say we wanted to make something in which we wanted the bacteria to proliferate, but then there was bacteriophage, a virus entered and it ate upon that bacteria and we could not get the designated substance. Okay? So we were considering bacteriophages as nuisance causing organisms. But of late, because of the development of meat as well as meat packaging industry, because packaged meat has, the demand for packaged meat has increased. So meat production there can be presence of bacteria and we want this bacteria to die. So we will introduce a virus which can feed on this bacteria so as to ensure that that meat does not contain any harm causing bacteria. So this virus, some viruses can be harm causing, some bacteriophages can cause harm, but then there can be other bacteriophages which are non-harm causing. So we will be introducing those viruses to the meat industry, to the meat, such that the meat does not get spoiled and it can be packed and its shelf life can be increased. Okay. So they are bacteriophages. Now talking about two categories of microbes, one is the harmful microbes and the second one is the useful microbes. Harmful microbes are basically disease causing. Disease causing matlab it will get in these microbes, chahe wo bacteria ho, fungi ho, virus ho, archaea ho, protozoa ho, this will enter into our body and will be causing some kind of infection. theek hai? Infectious disease matlab this can spread from one person to the other person very easily. This can spread through coughing, through mucus, through serum, anything can lead to spreading of this disease. In that case we will be calling that disease as infectious disease. If it does not spread, we call it as non-infectious diseases. And all of them, all the microbes which are harm causing, we call them as pathogens. Okay? The category of microbes which causes harm are known as pathogens. Now, some of the bacteria, some of the viruses we will be discussing. Basically, we will be discussing about some of the diseases and how is it related to some of the microbes and what is the spread of this disease, what are the cures for this disease. So, talking about one such disease is pneumonia. Pneumonia is caused by a bacterial disease. This is a bacterial disease. Okay? Otherwise, common cold, this is a viral disease. Common cold is a viral disease. Then we have Salmonella typhi. Salmonella typhi, as the name suggests, causes typhoid. This causes typhoid. This is the name of the bacteria. This is the name of a bacteria which causes typhoid. Typhoid itself is a waterborne disease. Okay. This bacteria, Salmonella typhi, will go and settle in the intestine. This will settle in the intestine and there is a test for checking whether the person is suffering from typhoid or not. This is known as Whittle test. Okay. So, Whittle test is carried out to check if a person is suffering from typhoid or not. Now, if a person is suffering from typhoid, it will this bacteria will be ejected in the human fecus. And this fecus will further spoil or introduce the bacteria to soil as well as water, and such a contaminated soil and water, if it is consumed by another person, they will also start suffering from typhoid. So, that is how it becomes infectious. It is infectious because this bacteria is present in human feces. Now, once this human feces meets soil as well as water, the bacteria enters into soil and water also, and from there the bacteria can spread, and if any other person consumes this contaminated water or soil, so then the new person will also start suffering from typhoid. Okay? So, this is how it is infectious. Then we have the case of malaria. Malaria is basically a protozoal disease. It is neither bacterial nor viral, it is a protozoal disease. But then the malaria has, usually we say ki for the previous diseases that we have studied, ki there are vectors. So, vectors can be mosquito, vectors can be sandfly, vectors can be pigs. Vector basically means any organism which is the carrier of a disease. Any organism which is the carrier of a disease is known as a vector. Okay. So, in the case of malaria, the vector itself is female Anopheles, which is a mosquito. Female Anopheles, which is a mosquito. Okay? And this female Anopheles will carry that malarial protozoa in their body, and once they bite somebody, when a mosquito bites another human, sporites are injected with bite. So, once the organism, human beings are infected by or bitten by a female Anopheles mosquito, then the malarial protozoa will be introduced inside the body. So, the parasite reaches the liver through blood, via blood this parasite is able to reach up to liver. As soon as it reaches up to liver, then they will reproduce and proliferate from liver, and from liver it will get transported into different parts of the body. Okay? So, now the person is suffering. Once it starts proliferating, this is the check that if the malarial protozoa is found in liver and has started to proliferate, then the person starts suffering from malaria. Okay? So at this stage we have detected that the person has started suffering from malaria. From here the malarial protozoa will proliferate, multiply, reproduce and spread to other parts of the body, okay, and releasing into blood. So this proliferated reproduced malarial protozoa will be reintroduced into blood, and from here it will start getting spreading to other parts of the body, okay. At RBCs it will also undergo sexual reproduction, okay. And then this female mosquito, this will be present in because a person is suffering from malaria and has been transported, malarial protozoa has been transported by blood, this will reach up to hand also. And if we have any other female anopheles which is not carrying malaria also, they will get this malarial gametes, they will get this malarial protozoa in their body, okay. And from here this new mosquito will start carrying the malarial protozoa with itself, and if they go and infect any other person, they will start suffering from malarial protozoa also. So if we can understand this, at this stage we understand that it is the mosquito which is the host, because here mosquito was carrying, it took the malarial protozoa from one organism and shifted, and it bite and it has bitten someone else who was not suffering from malaria. But in this case it is the human being which is the host, okay. In this case it is the human being which is leading to proliferation of malarial protozoa. So malaria ke case mein what happens, or this also happens in case of dengue and chikungunya and others. So what happens is, if a person is suffering from malaria, okay, and the first mosquito dropped the malarial protozoa in the human body, and from human body it entered into liver mein proliferation occurred, it entered into other parts of the body, and again at RBC it underwent a sexual reproduction, okay, at RBC, and then here also it was undergoing proliferation. Now the person is suffering from malaria and another mosquito has bitten this person. If this mosquito goes and bites any other person, then the person will start suffering from malaria. This is how malaria is infectious or contagious, because some part of it it is the malarial protozoa which is the host, it is the vector is mosquito, whereas the other half it is the human beings who are the vectors or transfers of they are the ones who are responsible for transferring the malarial protozoa, okay. So that is something special related to malaria as well as Aedes aegypti and chikungunya, okay. But then we have some good microbes also, we have some microbes which are helpful to us. One is an antibiotic, okay. Antibiotics may be, we make use of another bacteria, another organism which will be killing upon or eating upon the existing bacteria. Antibiotics will kill bacteria, and how do we make antibiotics is by making use of microorganisms itself, okay. So that is how it can be useful. In industrial production also we make use of various microbes. For example, yeast is used to make bread, okay. Then we have lactobacillus which is useful for curdling of milk. So to get a milk, some amount of milk to get converted into curd, we make use of lactobacillus. Lactobacillus is also present in human gut, okay, and this bacteria in human gut is also having the same task. They are responsible for digestion of milk protein, okay. Then we have Acetobacter aceti. This is used in formation of acetic acid, which is also commonly known as vinegar. So industrial production of vinegar we are making use of a bacteria called Acetobacter aceti, okay. So this is a bacteria, this is a bacteria, this is a yeast, okay, or basically fungi, okay. Then fungal lipase also has so many industrial applications. It is used in manufacturing, of textile, of leather, okay. So many areas where we are making use of fungal lipase, okay. Then fungi is also used as an immunosuppressant. So some of the human beings who have undergone organ transplant, they will have to consume an immunosuppressant because this immunosuppressant, because that they have taken an organ from a foreign body. Now our body understands that this organ does not belong to me, okay? So they will try to kill this, because for them this is not an organ, for my body this is a foreign particle, and it is important for the body's immune system to kill this foreign particle. So to kill them, we will introduce an immunosuppressant. To ensure that they don't kill this organ, we will introduce an immunosuppressant which is also made using fungi. Okay? Then we have streptokinase. Streptokinase is another bacteria. This bacteria is used for clot buster. So streptokinase is used when a person suffers from heart attack. So there has been some kind of blood clot which is not allowing the blood flow through vessels to take place. Blood vessels में blood flow नहीं हो पा रहा है, वो block हो जा रहा है because there is a blood clot. So what do we do is we will introduce this bacteria into the human body and they will go and dissolve the blood clot itself. So आराम से blood वापस flow करने लगता है, ठीक है? So streptokinase is used as an immediate measure for a heart attack victim. Okay. Then in terms of useful microbes, we also have we make use of microbes in fermentation also, ठीक है? In fermentation we make use of for example milk का curd में convert होना, या इडली and डोसा का batter के formation में भी we make use of bacteria, ठीक है? हम useful microbes में cheese formation के लिए भी we make use of bacteria. So have you observed कि cheese में there are big holes, French cheese when they show that cheese block, they are having big holes. That is because the bacteria which was staying there has released carbon dioxide, and it is these carbon dioxide holes which are present there. So it is the same bacteria is used in formation of cheese. Yeast is used in formation of sugar to alcohol. So alcohol formation में we introduce a yeast to sugar, ठीक है? We also make use of yeast to convert or create bread, ठीक है? Then bacteria are also used in sewage treatment. In sewage treatment we will introduce bacteria and allow this slow decomposition to take place. Because of that decomposition, all the organic matter will be decomposed and it will settle down at the bottom of the tank, and top of the tank say water we will remove, such that the lower part of the tank contains impure water, upper part of the tank contains pure water. So in sewage treatment also we make use of bacteria, ठीक है? Then we make use of bacteria in biogas production also. Biogas production में what do we do is organic sewage को, organic waste, domestic waste को we will cluster together, put together at one place, ठीक है? And introduce the bacteria at this point. Once you introduce the bacteria at this point, it will slowly lead to decomposition. Now because this is an organic waste, it usually leads to release of methane plus carbon dioxide plus water, ठीक है? And this is an anaerobic bacteria, ठीक है? This anaerobic bacteria, this set is also known as methanogens. Methanogens मतलब the one that releases methane. So because it is releasing methane plus carbon dioxide plus water, so this set of bacteria we call as methanogens, who are anaerobic bacteria, ठीक है? So anaerobic bacteria are used in biogas production. The methanogens are also present in rumens of cattle. Rumen is an organ within the cattle body which is responsible for cellulose digestion. They are responsible for cellulose digestion. Okay. Now because they are responsible for cellulose digestion, so whatever animals are eating through grasses or leaves, they contain a huge amount of cellulose. This can be easily decomposed at the site of rumen of cattle. Who does this is the methanogens. That is why cattle's के emissions में, cattle's के excreta में there is huge amount of methane gas which is released. Methane gas is released because it is a methanogen. It is an anaerobic bacteria which is living inside the rumen of cattle and is helpful in dissolution of cellulose. That results in methane based emissions from animals or cattle. ठीक है? Then we have some useful bacteria in biocontrol of pest also. So we make use of bacteria in reducing the pest infection on crops also. So agricultural fields mein we will be introducing bacteria such that this bacteria will ensure that the pest will not remain. Okay? For example, Bt bacteria. Bt bacteria, this we have already introduced. There is a genetically modified crop, genetically modified cotton, wherein we are making use of the genes of Bt bacteria, because this the gene is responsible for killing the insect larva. Okay? Because it kills the insect larva, so we will be able to, the crop that we have made, GM cotton that we have made, in this we have introduced Bt bacteria ka genes. Bt bacteria ka genes is responsible for killing the larva. Now the crop itself becomes resistant to larva or pest infections. Okay? So that is how it can be used in biocontrol of pest. But the idea is biocontrol of pest by using microorganisms, we make sure that we have this understanding that pests cannot be completely removed, but to reduce their presence or to keep the numbers in check, we make use of biocontrol agents such as bacteria, fungi, whatever it is, whoever is responsible for killing out of the pest. Okay? So they work on the principle of natural predation. Natural predation matlab there is something provided by nature, be it bacteria, virus, protozoa, fungi, anything that has been provided by nature. This will be responsible for eating up on the pest. Okay? So the idea is that I cannot remove all the pest, but I can definitely keep a check on all of this through natural predation. By natural processes, this bacteria will be able to kill out on the pest. Okay? Then microbes can also be used. This was a pesticide. So by microbes being used as a pest, so they will be called as a pesticide because they are stopping the pest control. They are stopping the pest proliferation. But biofertilizer matlab they will have to catalyze the production. Agriculture mein if you are adding a fertilizer, you are catalyzing the biological productivity. You are trying to improve the resources you are getting from the plant. Okay? So from plants we can increase the productivity by making use of not only urea based, DAP based or MOP based fertilizer, but biofertilizer also, matlab something that has been provided by nature. So biofertilizer may we have one bacteria which is responsible for binding nitrogen to leguminous plants. Leguminous plants ke root nodules mein we have rhizobium. Okay? Root nodules mein we have rhizobium. Rhizobium is a bacteria which is responsible for binding the atmospheric nitrogen to the soil. So they will be naturally adding nitrogen to the soil. The same task which is being carried out by urea is done by naturally through rhizobium bacteria. Okay? Then we have cyanobacteria. Cyanobacteria will be responsible for binding atmospheric nitrogen to water. Okay? Cyanobacteria will be responsible for binding atmospheric nitrogen to water. So they are usually present in water. So where should they be used? In cultivation of paddy. Okay? Then we also make use of a fungi called mycorrhizae. Usually we don't expect UPSC to be asking the name of the bacteria or fungi or virus until and unless it has been running in news. Okay? So we have skipped out on the names of bacteria because that can be confusing. If it is easy to remember, for example Acetobacter aceti, it is easy to remember because aceti is mentioned, so it will be responsible for formation of acetic acid. Or lactobacillus, so that means something related to lactose matlab milk. Okay? So that for that we have mentioned the name of the bacteria or virus or fungi, but for others we have not mentioned it because it may cause confusion. Okay? So talking about another microbes which is fungi, this is known as mycorrhizae. Mycorrhiza is also responsible for binding aeration of the soil. Okay? So these are also some of the useful microbes. Now talking about, so we spoke about harmful microbes. Harmful microbes will cause some kind of diseases amongst us. Okay? If you have to stop the occurrence of disease, so there should be some kind of natural immunity built up within your body. Okay? Immunity basically refers to the resistance that we are observing, the resistance from our body to not allow a foreign body to enter into us. Okay? So naturally, but nature itself has given us some of the capabilities that if any foreign body enters into our body, they will try to push it out, okay? Or try to kill it. They will either try to kill it or remove them or try to push them back. Okay? So, immunity refers to the capability of any organism to protect itself from any danger coming from foreign body. Okay? Now, this immunity can be an innate immunity. Innate immunity matlab something that we have received since birth. Matlab this is naturally present because we are living organisms. So, this is naturally present in us. So, it can be innate immunity, or another form of immunity can be an acquired immunity. Now, what is an acquired immunity? Acquired immunity matlab the person has acquired some kind of immunity against a foreign body. Okay? What is the meaning of it and how do we proceed with acquired immunity, that we will study. But first let us finish off innate immunity. Innate immunity itself is of 4 types. Okay? One is the physical barrier. So, skin is an example of a innate immunity because we are born as a human being or any organism. So, we have skin which will protect our body, which will protect the internal organs of body from any foreign particle. Okay? That is one physical barrier. Second is a physiological barrier. Physiological barrier matlab acids in stomach. This is an example of hydrochloric acid. Hydrochloric acid having pH close to 1 or 2, in the range between 1 to 2 is the pH of hydrochloric acid, which is released, secreted in human gut, stomach. Okay? Human gut may we release hydrochloric acid so that all the bacteria, pathogens, microbes that are present here which are harmful can be killed. But then we have good bacteria which are able to survive in such low pH conditions also, which was the second statement or third statement I think, or the question that was asked. There can be some microbes which can live in a pH condition of 2 to 3 also. Okay? So, there are certain bacteria which can survive such low pH of HCl also. Which is present in human gut. Okay? So, this is a psychological, physiological barrier. Physiological barrier matlab we have something inside, for example, saliva, lysosomes, something inside, saliva in our mouth is also having acidic pH. They are also responsible for killing out on the microbes which are entering into our mouth. So, saliva in mouth or hydrochloric acid in stomach can be an example of psychological barrier. Then we have a cellular barrier. Cellular barrier matlab there will be something which is present in the cells also. For example, our blood consists of red blood cells plus white blood cells plus platelets and plasma majorly. These are the 4 major components. Okay? Amongst them, this is the one which is responsible for providing us with immunity. So, there are so many components within white blood cells consisting of neutrophils, monocytes, B lymphocytes, T lymphocytes, lymphocytes basically. So, what is the task of WBC? This is known as phagocytosis. Phagocytosis matlab they will eat up, let's say WBC went somewhere and found that there is a foreign body sitting here. So, they will eat up that foreign body, that is phagocytosis, which is being carried out by WBC. So, their barrier matlab there is some cell in human body which is responsible for protection from any kind of foreign particles. Okay? Then we have in immunity, we also have a cytokine barrier. Cytokine barrier, for example, interferons. So, what are cytokine barriers? Let's say ki one cell okay, is infected by a foreign body. Okay? Let's say as soon as this cell is infected by a foreign body, they will create some kind of emissions. Okay? This emissions will ensure that this virus they will not allow to proliferate outside this cell. That is their intention. Whether the virus is more powerful or cells emissions are more powerful, that will determine whether the virus has been able to proliferate or not. But as soon as a cell is infected by a foreign body, okay, a foreign body, so this cell will start secreting interferons so as to ensure that this virus do not proliferate to other cells also. So, that is the another kind of innate immunity. Okay. But apart from innate immunity, we also have acquired immunity. What is an acquired immunity? Acquired immunity basically means this is pathogen specific. Pathogen specific matlab this is acquired. Let's say I acquired today with respect to a virus. Tomorrow I will acquire immunity with respect to bacteria. Day after tomorrow I will acquire immunity with respect to fungi. So, depending upon what is the pathogen, immunity will be acquired. Okay? and this is also characterized by memory. Characterized by memory matlab the cells, the human body will try to remember and recall, did I encounter this enemy earlier anytime? If I have encountered, then I will try to re-emit the same chemicals again, so as to ensure that because I know what is the cure against this foreign body, okay? Because I know that what is the cure, so I will try to emit the same let us say hormones, antibody, antigen, whatever it is, we will try to emit the same chemical substance so as to kill this foreign body, okay? So this is characterized by memory, so they will try to recall if we have encountered this kind of enemy earlier also, okay? Now, acquired immunity will be initially primary. Primary matlab when I have met with this foreign body for the first time. For example, the first infection of virus to my body, that is the primary response. Okay? Primary response matlab my body will try to fight against this virus. Okay? Let us say ki a virus entered into my body, I will first try killing it using gun. ठीक है, नहीं काम किया, okay, I'll try killing it using sword, नहीं काम किया, okay, I'll try to make use of a bomb, ठीक है, नहीं काम किया, so I'll try to make use of a missile and try to kill this, ठीक है. Now depending upon whether the virus is very powerful or not, it will have some kind of effect in my body. But then there is a primary response because I don't know which machine will work against this foreign body, ठीक है. So that is primary response. But secondary response is much more powerful. Kyunki now if I meet the virus once again, I know ki sword works against this enemy. So I will not make use of bombs and all that. As soon as this enemy enters into my body, I will make use of sword and kill that body, that foreign body. Okay? So that is the secondary response. This is much more powerful as compared to this one. Kyunki here you are aware about the enemy. Okay? Now, acquired immunity itself can be humoral immune response or cell mediated immune response. Humoral immune response matlab ki this is coming from blood, and that is why the term humoral matlab something related to blood. Okay? Within blood we have B lymphocyte. Okay? Where is B lymphocyte coming from? We remember that blood has components called red blood cells, white blood cells, plasma and platelets. So talking about white blood cells, they have this can be categorized into granular cells or agranular cells. Agranular matlab there will be no granules present here. Okay? If it is granular, it can be neutrophil, eosinophil or basophil. Neutrophil ka task is phagocytosis. So as soon as an organism comes, they will try to eat it up and consume it and remove it. Okay? Parasitic infections se fight karne ke liye we have eosinophils and allergic inflammatory reactions. So for example, some foreign body, let's say you might have often encountered this ki a wood chip enters into the body and remains there. Okay? Ya anything spine enters into the body and remains there. Okay? So there will be a swelling here, swelling is some inflammation and some allergic reactions that is coming out of basophils. Okay? Then agranular matlab they will produce some kind of immune response. From here this lymphocytes emit either B lymphocyte, T lymphocyte or natural killer cells. Okay? Then we have monocyte also. The monocytes are responsible for killing off bacteria, viruses and fungi. Okay? So neutrophils and monocytes they will undertake direct phagocytosis. Okay? They will directly consume it or kill it. Okay? Neutrophils will kill it, neutrophils will consume it, monocyte will kill it. Okay? Lymphocyte will produce the immune response. See, this is the organism, foreign body. So you try whether this will work or this will work or this will work. No, or otherwise secondary response may we know that this works. So that is what we will be using against this organism. Okay? That is the task of lymphocyte. Basophils is responsible for inflammation, okay? And eosins are responsible for parasitic fighting against the parasitic infections. So such all of this because it is present in blood, so we call this as humoral immune response. Okay? Humoral immune response is coming from the B lymphocyte. Okay? And what they will do is a foreign body ko we call it as antigen. Okay? So our body will produce some kind of antibody to kill this antigen. Okay? So antibody will be produced in blood so that this antigen will be killed at the spot itself. Okay? Then cell mediated immune response, because all of this immune response is present in blood, so we call this as humoral immune response. Now because this is taking place at the cell, at any other cell, we call this as cell mediated immune response. Who is responsible for this? It is the T lymphocyte. Okay? They themselves are not responsible for killing, but they will go and inform B lymphocyte that you have to kill. Okay? Now cell mediated immune response can be, example to this can be rejection of graft cell. So we discussed about this, ki there is a let's say there is an organ transplant and a new organ has been introduced in human body. Okay? So because my body now knows that this is a new organ, this is not belonging to my body, so I will go and kill this organ. Okay? All of this are responsible for killing this or eating this organ, let's say kidney transplant. Okay? So what do we do is, so who is responsible for killing this? It is the T lymphocyte, or that they are the ones who are responsible for rejection of graft cell. This is a graft cell and they will reject this. T lymphocyte will reject this. No, you do not belong to my body. So T lymphocyte will go and inform B lymphocyte that you need to go and kill this graft cell. Okay? What do we do is, we will put up immunosuppressant. So this immune response we will try to suppress, such that this is a new cell which should not be killed. Okay? Although this is a new organ to your body, but this should not be killed. So we will make the person consume some kind of immunosuppressant such that they do not go and kill that organism. Okay? So that is the intention of a cell mediated, immune response, ki any kind of a new graft cell, matlab they will try to go and kill that cell. So we call this as a cell mediated immune response. And who is responsible for this? It is the T lymphocyte. Okay. Now with that we come to the end of this video. We will continue our discussion related to another question of previous year's 2023 paper itself. I wish you all the very best and thank you so much.